Hello friends, today we are going to start topic of plant physiology, that is phytohormone. In this video we will discuss about features of phytohormone and then types of phytohormone. We will mainly focus on auxin. We will discuss about the discovery of auxin through various experiments. Then we will discuss about physiological effect of auxin. We will discuss about auxin biosynthesis, and at last we will discuss about auxin signaling pathway. So keep watching. Let us start with phytohormone. So first of all, let us see what is plant growth. It is irreversible change in size. That means the change in size is permanent or it cannot be reversed. And this change in size may be increase in length or volume. And this growth is considered as increase in dry weight. Wet weight is not considered because it keeps on changing. So how plant growth is controlled? These are controlled or regulated by phytohormones, which are also known as plant hormones or plant growth regulator. Let us see its definition. These plant hormones are naturally produced organic substances and these are produced in higher plants. These control the growth or physiological functions in minute amount. That means in order to control the growth or physiological functions, small amount of phytohormones are required. Now let us see the features of phytohormones. These are small organic compounds. These are synthesized by specific plant cells or tissues. These are active in low concentration. It means only small amount of phytohormones are required in order to control the plant growth. Promote or inhibit growth or developmental responses. Site of function is remote from site of production. That means the site where it shows the function is separate from the site where it is produced. Now the types of plant hormones. We will discuss about 5 major types of plant hormones. Auxins, gibberellins, cytokinins, abscisic acid and ethylene. Let us discuss them one by one. Auxin. Name is given from the Greek word auxin, that means to increase. It is the first plant hormone that was discovered. It was discovered in the last quarter of 19th century and it was successfully isolated by Fritz Wendt from the tip of Avena sativa coleoptiles, which is oats. Now let us discuss some of the experiments that were conducted which led to the discovery of auxin hormone. First of all, in 1880, Darwin did his experiment. He took the coleoptile of grass and exposed it with unilateral light. What he saw that these coleoptile bent towards the light. Later on, he covered the tip of the coleoptile with the tin foil and then exposed to the unilateral light. At this time, no curvature or bending was seen. So what it was concluded that tip of the coleoptile senses light and also that certain stimulus is transmitted from the tip of the coleoptile to the lower region, which causes the bending of the coleoptile in the direction of the light. But when the tip is covered with tin foil, light is not sensed. In 1910, Boysen and Jensen cut the tip of the coleoptile and then replaced it, keeping a gelatine plate, and was exposed with the unilateral light. Again, the bending of the coleoptile was seen. So what he concluded, on the basis of which that the stimulus that Darwin has mentioned is actually material substance which diffuses through the gelatin from the tip of the coleoptile to the lower part. In 1919, Pal, he cut the tip of the coleoptile and placed it asymmetrically. Then it was kept in the dark. but still the coleoptile shows the curvature or a bend. So what it was concluded that these material substances that were produced from the tip, when these diffuses to the lower part, then it causes the growth of the lower part. Here this is placed asymmetrically, so these diffuses to the lower part or transmitted to the lower part only in this region. As a result of which this region grows more compared to this region. So there is asymmetry in the growth of these two regions. As a result of which this region grows more compared to this, and hence it bends or it shows the curvature. And this also explains the bending of the coleoptile in the direction of the light when it is exposed to unilateral light. Because in the presence of the unilateral light, there is more concentration of auxin in the shaded region compared to the illuminated region, and hence more growth or elongation of this region compared to this region, as a result of which there is asymmetric growth and coleoptile bend towards the direction of light, or the curvature is seen. But when light is given to the coleoptile equally in all directions, then there is no asymmetric growth, but the growth is symmetric, and hence in that case there is no bending of coleoptile or curvature is not seen. Later on in 1928, Wendt cut the tip of the coleoptile and placed them on the gelatin or agar block. Then these agar block were placed on this coleoptile asymmetrically, and then these were placed in dark. It was seen that this shows the curvature. That means the material substance which causes the growth of the lower part has diffused from the tip of the coleoptile to the agar block, and when it was placed, then it diffuses from agar block to the lower region. Since it is placed asymmetrically, this region receives this substance, but this region doesn't receive the substance. Hence, this region shows more growth compared to this, and this results in the bending or the curvature in the coleoptile. And this led to the discovery of auxin, and on the basis of this experiment, Wendt was able to isolate auxin from the Avena sativa coleoptile, that is the oats coleoptile. Some of the naturally occurring auxins are indole-3-acetic acid IAA. This is its chemical structure. Indole-3-butyric acid, that is IBA. This is its chemical structure. Phenyl acetic acid is also naturally occurring auxins. There are more than 200 auxin analogues also present, and these have the common feature, that is these have the unsaturated planar ring structure. This is the planar ring structure and a carboxyl side chain. This is carboxyl side chain. Now let us see some of the physiological effects of auxins. First is cell elongation. It stimulates elongation or expansion of cells in shoot. Cell division. It stimulates cell division in cambium and also in tissue culture with cytokinins. That is, in the tissue culture, in addition to cytokinins, when auxin is also added, then it results in the cell division. Cell differentiation. It stimulates cell differentiation of phloem and xylem. Root initiation. It stimulates root initiation on stem cuttings and also lateral root in tissue culture. Apical dominance. When there is apical bud present, it inhibits the growth of the lateral bud. This is known as apical dominance. When the apical bud is removed, then lateral buds grow. Flowering. It promotes flowering in bromeliads and femaleness in dioecious flowers. Next is callus formation. In many tissue culture, by the addition of auxin. So in many tissue culture, when auxin is added, it results in the formation of callus. What is callus? It is undifferentiated mass of cell. When auxin is added in the tissue culture, then it results in the division of the cell, and hence undifferentiated mass of cell is formed, which is known as callus formation. Tropic response, that is bending to gravity, known as geotropism, and bending to light, known as phototropism. This phototropism we have already seen in the experiments that were conducted in the discovery of auxin. Parthenocarpy, auxin-induced parthenocarpic fruits. What are parthenocarpic fruits? Fruits that are produced without fertilization of ovule. It occurs naturally but can be induced artificially. Now let us discuss biosynthesis of auxin. Sites for the auxin synthesis are shoot apical meristem, young leaves, developing fruits and seeds. Now the pathways of auxin biosynthesis. There are two pathways of auxin biosynthesis. First is tryptophan independent pathway, TIP, and the second is tryptophan dependent pathway, that is TDP. Tryptophan independent pathway does not involve tryptophan and its mechanism is not well known. So in this video, we will mainly discuss about tryptophan dependent pathway. In this pathway, the precursor is tryptophan. It is synthesized by shikimate pathway in plants, but this pathway is not present in animals. Now in the tryptophan dependent pathway, four types or four possibilities are there. One is IPA, that is Indole-3-Pyruvic acid pathway. Second is TAM, that is tryptamine pathway. And third is IAN, Indole-3-Acetonitrile pathway. Fourth is IAM, Indole-3-Acetamide pathway. In this, the first three pathway, IPA pathway, TAM pathway and IAN pathway, these are present in plants or these are used in plants. But this IAM pathway, it is used in bacteria. Example, Agrobacterium. Now let us discuss about these pathways in detail one by one. First is IPA pathway. Tryptophan undergoes deamination in the presence of the enzyme tryptophan transaminase and it is converted into Indole-3-Pyruvic acid, that is IPA. Then this IPA undergoes decarboxylation in the presence of the enzyme IPA decarboxylase and it is converted into Indole-3-acetaldehyde. This Indole-3-acetaldehyde then undergoes dehydrogenation in the presence of the enzyme Indole-3-acetaldehyde dehydrogenase and is converted into Indole-3-acetic acid, that is naturally occurring auxin. So here the first intermediate of the pathway is IPA, hence it is known as IPA pathway. Now second is TAM pathway. Here tryptophan undergoes decarboxylation and it is converted into tryptamine, in short TAM. Then this undergoes deamination and is converted into Indole-3-acetaldehyde. This Indole-3-acetaldehyde undergoes dehydrogenation in the presence of enzyme Indole-3-acetaldehyde dehydrogenase and is converted into Indole-3-acetic acid. Here the first intermediate is tryptamine or TAM, hence it is known as TAM pathway. Third is Indole-3-Acetonitrile IAN pathway. Here tryptophan in the presence of certain enzyme is converted into Indole-3-Acetonitrile IAN. This is then converted into Indole-3-acetic acid in the presence of the enzyme nitrulase. Here the first intermediate of the pathway is Indole-3-Acetonitrile, hence it is known as IAN pathway. Now IAM pathway. Tryptophan in the presence of the enzyme monooxygenase is converted into indole-3-acetamide IAM. This is then converted into indole-3-acetic acid. So here first intermediate is IAM and hence it is known as Iam pathway. This type of the pathway is used in bacteria. Example, agrobacterium. We can show all these pathways briefly here. This is the first pathway, this is the second pathway, this is the third pathway, and this is the fourth pathway. All starts with tryptophan. In the first pathway, the first intermediate is IPA. Here the first intermediate is TAM. Here it is IAN, and in fourth it is IAM. And the product that is obtained through all the pathways is IAA, indole-3-acetic acid, which is naturally occurring auxin. Now let us see auxin transport. So transport of auxin occurs unidirectionally. That is, it is predominantly polar. It is an active process because here metabolic energy is required. And why metabolic energy is required? Because it is not simple diffusion. It can occur against concentration gradient also, and it involves activity of living cell. Hence, energy is required, so it is an active process. In shoot, it is polar and basipetal. That is, transport of auxin takes place from shoot tip to roots, or in other words, apex to base. And it occurs through vascular parenchyma tissue. In roots, it is polar but equipetal, that is from base to apex. Polar transport is inhibited under anaerobic condition. It is also temperature sensitive. Some of the metabolic inhibitors of auxin transport are 2,3,5-triiodobenzoic acid, TBA, and naphthyl-thalamic acid, NPA. Now let us discuss about auxin signaling pathway. We already know that auxin is a crucial plant hormone. It controls many aspects of plant growth and development. Or in other words, it has many physiological effects. And these physiological effects are brought about by transcription regulation. Or in other words, by transcription regulation, it brings about physiological effects. Auxin is responsible for changes in gene expression when a plant cell comes in contact with it, or when a plant cell comes in contact with auxin, it is responsible for changes in gene expression, and this results in upregulation or downregulation of genes. Any signaling pathway has certain key components. Similarly, auxin also has certain key components. Let us discuss them one by one. First is ligand. It is signaling molecule. Here it is auxin. Receptor, which binds with ligand. Here it is TIR1AFB family of F-box proteins. It is known as the auxin receptor. So it acts as auxin receptor plus it has E3 ligase activity. That is, it attaches polyubiquitin to target protein for its destruction. Third is transcription factor. It helps in transcribing necessary genes. Here it is ARF, known as auxin response factor. Then fourth is inhibitor. It suppresses the ability of ARF to enhance gene expression. Here it is AUX/IAA. Now let us see how the entire signaling takes place. There are two possibilities or two situations. First is when auxin is absent. Second, when auxin is present. So let us discuss them one by one. When auxin is absent, AUX/IAA, which is inhibitor, bind to and suppress the transcription activity of ARFs, which is transcription factor. As a result of which transcription is suppressed and hence auxin signaling is off. So when auxin is absent, it does not bind to TIR1 F-box complex. Inhibitor AUX/IAA binds with ARFs, which is transcription factor. It suppresses or inhibits its activity. Auxin signaling is off. So how it happens? When the AUX-IAA is at the promoter with ARF, then this AUX-IAA recruits other factors to make modification to the DNA structure, as a result of which ARF is not able to transcribe the genes and hence auxin signaling is off. Let us understand this with the help of the figure. Auxin is absent. Hence, auxin doesn't bind to this receptor. Inhibitor AUX/IAA comes and binds with ARF. It suppresses the activity of the transcription factor, that is ARF. As a result of which auxin response gene is not transcribed and we say that auxin signaling is off. Here AUX-IAA and ARF forms heterodimer. Second situation, when auxin is present, auxin forms molecular glue between receptor and AUX/IAA, that is inhibitor. Receptor also acts as ubiquitin ligase and leads to polyubiquitination of inhibitor AUX/IAA. Once inhibitor is tagged with polyubiquitin, then it is degraded by 26S proteasome. Now with the degradation of inhibitor, ARF transcription factor is free. Hence it transcribes the genes and we say that transcription of genes is on or the signaling is on. So auxin is present, it binds to receptor. This receptor then binds with inhibitor AUX/IAA. As a result of which the transcription factor ARF is free and hence the transcription is on or there is auxin-induced gene expression. Let us understand this with the figure. Auxin is present. Auxin comes and binds with the auxin receptor. It acts as the molecular glue between AUX-IAA and receptor. This receptor also has the E3 ligase activity. It tags AUX-IAA with polyubiquitin. Then this inhibitor, which is tagged with polyubiquitin, undergoes proteolysis by 26S proteasome and hence it is degraded. In the absence of this inhibitor, this ARF is free. It forms homodimer and then results in the transcription of auxin response genes. We say that transcription is on. There is auxin-induced gene expression. But if this is not regulated, then the transcription goes on. And this will result in the wastage of materials and energy. So there should be certain regulatory system. Among the genes that are transcribed or among the auxin response genes, there are certain genes that encodes for AUX-IAA, which is inhibitor of transcription factor, ARF. So when it is encoded, then this inhibitor comes and binds with the transcription factor and suppresses its activity. So it creates a negative feedback regulatory system, and in this way it is regulated. So this is all for today's video. In the next video, we will start with another plant hormone, gibberellin. So stay tuned. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.